this session. So Nima, yeah, perfect.
First of all, thank you,
Thank
Dr.
you.
Akbar, for a great presentation, and I'm happy to announce our next speaker, Dr. Hilal Rather. He has done his PhD in India Central University, Gujarat. Back there, he was doing his PhD working on biomaterial-based drug delivery system for bone regeneration, and now he's a research associate for postdoctoral research at Wake Forest School of Medicine. And we are happy that we have him today to talk about prostate cancer-derived smaller EVs promoting neuroinflammation and cognitive impairment. Dr. Hilal, the stage is yours. Thanks for
Hi Nima, can you hear me?
Yes.
Thank you for the introduction. Yeah, I'm just trying to load my presentation somehow. I'm sorry because of the time differences. I have joined little late, and then now I was trying to figure out how. Yeah, I can see. I can share my screen. Give me a second. It should be there up and running in a second. Can you see it now?
Yes.
Okay. And one more second. I will. I'm unable to figure out where I can. Oh yeah, I think I figured it out.
All good, thank you.
Is it visible? Okay.
Yes, awesome.
Okay, great. Yeah, and thank you so much, and thank you everyone for joining us today. Hi, as introduced, I am Hilal Rather. I did my PhD in India, and then I moved to United States. I did postdoctoral research in Wake Forest University, which I'll be sharing today. But presently, I'm working in Duke University, which is again in North Carolina States. So today I'll be talking about prostate cancer derived smaller extracellular vesicles promote neuroinflammation and cognitive impairments. So starting with a brief introduction about the prostate cancer, it's the second most cancer in men in United States. More than 0.26 million cases were reported for the year 2022. 1 among 8 men throughout the world will be diagnosed with prostate cancer throughout their lifetime. However, only 1 among 48 men will die of prostate cancer. That is predicted by a Cancer Society of America. So although these numbers are not really high, in fact, more than 3.1 million men in the United States who have been diagnosed with prostate cancer at some time point are still alive today. However, their lifestyle is pretty much affected due to illness or treatments. They experience issues such as erectile dysfunction, urinary incontinence, fatigue, pain, and this forced lifestyle change could lead to psychological distress. And lately, impaired cognition and dementia, as well as Alzheimer's disease, is also linked with prostate cancer. So these are some reports which had highlighted dementia or Alzheimer's disease or any other psychiatric issues. So this study, it's a retrospective cohort study where they had more than 150,000 elderly men above 70 years of age, and they found that in the elderly men, androgen deprivation therapy, the diagnosis of Alzheimer's disease was higher as compared to the patients which were not treated with androgen deprivation therapy. And then all-case dementia was also higher. So other study was also reporting almost similar kind of results. They also showed that androgen deprivation therapy, it has higher prevalence of Alzheimer's disease or all-case dementia. Then one more study, they also showed the incidence of depression was almost double in the patients who were treated with androgen deprivation therapy. So in nutshell, although the androgen deprivation therapies, although they are majorly associated with dementia or depression, but even the patients which do not have undergone any androgen deprivation therapy can still develop dementia or depression. So our question was what could be the possible link between prostate cancer and some effects in the brain. So as a exosome or small extracellular vesicle, as a extracellular vesicle lab, we were mainly into finding the link through small extracellular vesicles. As most of you are already aware that these are the nano biomolecules, which are nano vehicles, which are in the range of approx 40 to 150 nanometer, and they are released by all cell types. These exosomes, they can be carried to and fro between the tissues and circulation. And interestingly, there are reports that even tumor-derived extracellular vesicles, although these study had shown brain tumor-derived extracellular vesicles, they could cross blood-brain barrier. And then loading of this cargo will depend upon the physiological state of the cell where these EVs are released from. Furthermore, in case of prostate cancer, EV loaded microRNA has been related the content has been reported to change with the castration when castration resistant prostate cancer, when it undergoes neuroendocrine differentiation, which is the higher grade prostate cancer. So overall, EV cargo could be useful to predict the link between prostate cancer and cognitive impairments. So to answer this, we use TRAMP mice, which is a transgenic mice model for prostate tumor. So these mice, we had around 50 to 54 weeks of age, and then we performed this water Morris maze test, which is a procedure to assess spatial learning and memory in the rodents. If you could see here, this is you put the mice or you put any rodent in this Morris water maze, and it is provided with the visual cues, and there's a hidden platform. So if the memory is not altered, so they should find this invisible platform in lesser time with the progression in the training. So we train the mice up to 5 days. If we could see, like, the first day and the last day, the mice should take lesser time to reach to the platform as compared to the first day. So if you could see here, the escape latency, which is the time taken to reach to that invisible platform. So the TRAMP mice, it took significantly more time to reach to that platform as compared to the control mice. So this indicates that altered learning behavior in these TRAMP mice, which could be an indicator of cognitive impairment due to prostate cancer. So further, we did a long-term potentiation analysis, which is one of the most intensively studied ex vivo model for learning and memory by measuring the synaptic plasticity. So the postsynaptic alterations has been kind of reported, and we see that in this is the control mice and this is the TRAMP mice, which has the prostate tumors. So you could see there's significantly altered long-term potentiation, and this shows the end time point, which also at the 90th minute of the experiment, it was significantly reduced in case of the TRAMP mice as compared to the control mice. So which also indicates or supports that these mice, they had cognitive impairments due to prostate tumor. So further, in case of the human or patients with Alzheimer's disease or dementia, cognitive impairments are reported to have elevated amyloid beta deposits. Therefore, C11 PET scan was performed, which is an analog for thioplomin-T, which selectively binds to the amyloid beta. So in case of the mice, when we injected this, we could see sagittal, coronal, or axial scans, PET scans. We could see significantly more amyloid beta deposits because of these C11 PIB scans, and this shows the overall brain quantification from overall brain. It was 3.65-fold higher than the control mice. So further, we validated this with the IAC analysis, IAC from hippocampus, cortex, and cerebellum. So this is the quantification where you could see significantly higher amyloid beta deposits in case of the TRAMP mice as compared to the control mice. Although there was there was trend, but it was not statistically significant in cortex and cerebellum. So as hippocampus has a major role in memory and learning, so that could be one of the reasons that the hippocampus was majorly affected. So as I said, we wanted to see or we were focused on smaller extracellular vesicles as a connecting link. So what we did is we isolated small extracellular vesicles from the TRAMP cell line, which is cell line isolated from these transgenic mice tumors, and then we isolated, and this is just the representative NTA of various batches, which represents the size below 200 nanometer. Then zeta potential analysis also shows the negative zeta potential of these, which is pretty much reported in the literature. Then with the tetraspinin analysis by flow cytometry and transmission electron microscopy by using immunogold labeling for CD9, CD63, and CD81, and you could see the expression by flow cytometry as well as transmission electron microscopy, which is just to characterize these smaller extracellular vesicles. So now the second question was that if these EVs they cause these effects in the brain, then whether they reach the brain, or they specifically target the brain, or they do some other organs as well. So for that, we did a biodistribution analysis. For this study, what we did is we took these TRAMP C1 cell EVs, then transfected these with C-Elgen microRNA 39, which was labeled with Cy5. This shows the successful transfection. This is control where we did not transfect with C-Elgen Cy5, and this shows around like co-positivity, like we shows like around 90% transfection, 89.57% to be precise. So when we injected these EVs, and after 24 hours, we sagged the mice, and we did imaging analysis, and we found that this is the control mice, and these are the C-Elgen transfected mice. I mean the C-Elgen transfected EV injected mice. So you could see in organs there was slight some signal was from liver, which is pretty much obvious. Most of the nanoparticles go to liver, and then but in case of the brain, you could see the C-Elgen 39 EVs were reaching to most part of the brain, hippocampus, cortex, cerebellum, and other parts as well. So now the next question was, as it was reported that blood-brain barrier these EVs can cross blood-brain barrier, but they can cross without disrupting the blood-brain barrier. So this experiment did not answer that question. So next we wanted to see whether the blood-brain barrier was intact or it was disrupted. So we did this experiment where we again injected unlabeled EVs, and then after 24 hours we injected sodium fluorescein dye. If the blood-brain barrier is disrupted, it will cross the blood-brain barrier. Otherwise, it will not cross. So this analysis where you could see the signaling from the mice which were injected with small extracellular vesicles. So we then homogenized these brains, and we did fluorescence measurements, and you could see significantly higher signaling here. So which kind of shows that this blood-brain barrier was disrupted in these mice. So further, we also checked this on in a I'm sorry, my presentation is somehow stuck. Yeah. So further, we checked this on transfer assay. We used microvascular endothelial cell line to represent a model for blood-brain barrier, and then after establishing the barrier by measuring when the tear measurements were stable, we treated these small extracellular vesicles at different concentrations, and then we collected. Then we treated the fitsidextron, and then collected the flow through or the release rate or the media from the lower chamber, and we then performed the analysis from that, and we could see that the fitsidextron intensity was significantly going up with the higher concentration, which also kind of indicates that the barrier was disrupted with the treatment of these extracellular vesicles. So now we had these. We also did inject these smaller extracellular vesicles at 10 microgram twice a week in the normal age-matched mice. So we started at around 23 weeks, and we we started around 32 weeks, and we injected for around 23 weeks until it was age-matched with the TRAMP mice. And then we observed that their long-term potentiation was also altered. If you could see the red ones are SEV injected mice. So also the 30-minute the end time point analysis also showed the same results. So which also kind of indicate that these EVs could cause the alteration in long-term potentiation. So now the question was, what is loaded in these EVs? So as like various cargo is reported with proteins or nucleic acids or any other thing. So we did non-coding small RNA sequencing because non-coding small RNAs are majorly reported to be loaded in these small extracellular vesicles, and we find that interestingly from the top hits we find these 4 microRNAs 125, 128, 146, and 222. These 4 microRNAs were highly enriched, and then we run an analysis, over-representation analysis using mere microRNA enriched analysis and annotation tool, and we find most of them had demonstrated the role in neurological disorders, as you could see here. So so we did further what we did is we collected the RNA from hippocampus, cortex, and cerebellum just to see if these microRNAs were enriched there and to validate our results. So we see most of these microRNAs 128, 146, and 222, they were significantly overexpressed or highly enriched in the hippocampus of FRAMP mice as compared to the control mice. Although cortex also had some trend, increasing trend, but it was statistically significant, and then cerebellum had a reverse trend, although it was again not statistically significant. We also did this microRNA analysis from visceral organs like heart, liver, lung, kidney, and spleen, but we did not see any significant difference there. So which was an indicator that specifically the hippocampus is getting altered, or these microRNAs are getting targeted to hippocampus by virtue of these small extracellular vesicles. So interestingly, we also observed that the tumors, if you could see the red ones, these are the TRAMP tumors. So we isolated RNA from there, and then we isolated from control lower urinary tract of the mice, from the control mice, and we see that the expression was decreased in tumor as compared to the control lower urinary tract. So one possible reason could be that selective loading of these microRNAs in small extracellular vesicles, which could lead to more enrichment in small extracellular vesicles as compared to the tumors. So as it was recently reported in one of the papers published in JEV, they have also showed that prostate cancer secreted prostate cancer secreted microRNAs, they were selectively highly enriched in small extracellular vesicles as compared to the cells. So that could be the reason. But to further validate it, we took a range of prostate cancer cell lines, D145, PC3, and 22RE1 sensitive and resistant, NIH H660 cell lines. These cell lines range from bone metastatic, brain metastatic, and then enzymatic resistant, and then the H660, these are neuroendocrine, which are the late stage prostate cancer cell lines, and the TRAMP1, which are mice prostate cancer cell lines. So you could see here the delta CT values, which represents as compared to the cells, which are like the black bars, and the SCVs, which are the red bars. You could see the delta CT of the red bars from the SCV. They were lower than as compared to the cells, which indicates, so the lower delta CT means more enrichment or more expression of these microRNAs. Except this, in the TRAMP, all other microRNAs from all the cells were highly enriched in the small extracellular vesicles as compared to the parental cells. So next we had these mice where we had injected small extracellular vesicles. We interestingly also find the same phenomena where except this microRNA 125, these 3 microRNAs 128, 146, and 222. If you see, the black one is from hippocampus, this is from red one is from cortex, and green one is from cerebellum. So the delta CT was lower in hippocampus, which means these microRNAs were highly enriched in hippocampus as compared to cerebellum and cortex. So next we performed the same microRNA analysis from the plasma of these mice. So we pulled out total EVs using precipitation method. We use ExoQuick for that, and then the total EVs. We performed this analysis where we could see that although there was not a significant difference, but there was an increasing trend in all 4 of these microRNAs from the plasma of these mice. Further, we also analyzed the clinical relevance of these smaller extracellular vesicle enriched microRNAs in patient-derived prostate cancer specific EVs. So we had samples from 9 patients, which showed which had Gleason score more than 7, and we collected prostate. We first did a total EV isolation using ExoQuick method, and then from there we use PSMA positive. We collected PSMA positive EVs, which is a prostate specific membrane antigen, which is highly expressed in terms of in case of prostate cancer as compared to the normal prostate. So this just shows that PSMA expression on these smaller cellular vesicles, and then these all 4 microRNAs, they were the detectable levels were there in the PSMA positive EVs. So which could which could correlate it with the clinical relevance of these small cellular extravesical enriched EVs in prostate cancer patients. So now the next question was that so far we had established that these prostate cancer secreted EVs, they could cross the blood-brain barrier, and they could load the microRNAs in the specifically in the hippocampus. So now the next question was how is the memory or their cognition is getting affected. So as brain, it's a heterogeneous organ. So we did analysis of neuronal markers, astrocyte markers, and microglia markers. This is our real-time PCR based, but we did not find any difference from neuronal markers or astrocyte markers. But we could see some difference in microglia markers, specifically M1 microglia, which are pro-inflammatory microglia. This is INOS expression. The red ones is the trap mice, and IL-6, which is again a pro-inflammatory marker. It was significantly higher in hippocampus specifically, interestingly. So we although like the M2 markers, they were slightly reduced in hippocampus, as you could see the arginase 1 and CD206, and there was not much difference in cortex and cerebellum of either the pro-inflammatory or anti-inflammatory markers. So we further wanted to validate if the IC analysis from hippocampus, cerebellum, cortex. We did IC analysis for IL-6, and we see some increasing trend, but it was not statistically significant. Next, we did as these microRNAs, they could regulate the expression of genes, and one microRNA can regulate multiple genes, and even multiple microRNAs in together can regulate a single gene. Therefore, we did an unbiased target gene analysis for all 4 of these microRNAs, and then we loaded top 50 targeted genes, and we did ingenuity pathway analysis. And interestingly, if you could see the predicted target pathways, this is the top 10 pathways. So 7 out of the top 10 pathways were associated with neuroinflammations. So as NF-kappa B activation is well established in signaling pathway of inflammation and neurodegeneration. So we did an NF-kappa B reporter assay. So we had this THP1, which is a monocyte cell line. We had the reporter NF-kappa B gene in them. So we treated these cells with smaller extracellular vascular, and for positive control, we used LPS. So as you could see, the trend was there, increasing trend of luminescence was there. So if it activates the NF-kappa B, there will be more luminescence. We collected the supernatant and we performed the luminescence detection there. And after 24 hours and 48 hours, it was significantly higher as compared to the control ones. So which was also indication that these EVs, they could induce the inflammatory response. So further, as again the question was that whether these EVs they could induce the inflammatory response, and because of whether it was actually because of these microRNAs. So what we did is we next inhibited or used anti-mirs and transfected these EVs with the anti-mir 125, 128, 146, and 222. We also did the combination for all 4 of these. As you could see, like this is just the cell culture control where we just treated PBS, and this is the positive control where there should be an obvious higher luminescence. And then this is the actual control for the transfection. You could see the luminescence has gone down with the transfection of these anti-mirs at different time points. We did 24 hour, 48 hour, 72, and up to 96 hour time point. So that was interesting to see that while inhibiting these microRNAs in the small extracellular vesicles, we could see the reverse trend in the inflammatory response. So as it's reported in if the microglia is getting activated, there could be some morphological change as well. So what we did is we also we had these BV2 and SIM anins, which are mice microglia cell lines. We treated this is the control PBS. This is treated with small extracellular vesicles. This is positive for M1 polarization LPS, and this is positive for M2 polarization IL4. So if you could, interestingly, we saw like there was a certain morphological change. The cells could, from the compact structure to the cuboidal or spindle shaped structure was observed. So that was again an indication that they could change the phenotype of the cells, these microglia cells. And next, the question was whether it was the phenotype was M1 or it was M2, which is pro-inflammatory or anti-inflammatory. So we did their marker analysis by real-time PCR. So we again seeded these cells and then treated with them with small extracellular vesicles and with positive and negative controls. And then we observed that cells treated with these small extracellular vesicles, they have more expression of M1 markers like INOS, IL-6, TNF alpha, and IL-1 beta. However, the M2 expression, which is M2 marker expression, which is arginase-1 and CD206, it was significantly reduced. So overall, to summarize this study, I would say that the prostate cancer secreted small extracellular vesicles could cross blood-brain barrier, and then they could load these specific microRNAs, which could cause M1 polarization of microglia, and which could further lead to cognitive impairments. So I would briefly like to acknowledge my lab, my mentor, Professor Gagandeep, Dr. Ashish Kumar, who is an assistant professor in the lab, Dr. Shalini Mishra, and Dr. Sam Al-Musa, who helped me with biodistribution studies, Susie Kin, who helped me with most of the animal studies, Ishin, who helped me with the plasma samples, as she has all the plasma repository, and also helped me with the transmission electron microscopy. Dr. Sangeeta with the anti-analysis, Dr. Mitu with cell culture studies, Yangin again with cell culture studies and some of the RNA studies, Hitanshi, who is still working on this project and and had also helped me with some IC studies, Jacob and other lab members as well. So I'd also like to acknowledge our collaborators who helped with different experiments and different instruments, and National Institute of Health for funding various projects in our lab. And thank you so much for giving me this opportunity, and thank you everyone for attending this talk today. And I'll be open for the questions.
Thanks, Dr. Hilal, for great presentation and detailed presentation. And also, and now I don't see any question in the chat box. We can wait just 1 minute. If nobody has any question, I can ask one. Oh, Carlos, please go ahead.
So, hi. Thanks for the quite comprehensive talk. A lot of data once again, and a lot of tools to study once again, which is really cool to validate points. I was curious about, because you showed that treating the animals with maybe I missed what was the administration route, but treating the animals with the EVs, it could permeabilize more the BBB, correct?
Yes.
Do you know the mechanism that is related to this increase of the permeabilization?
I mean, we have not, I mean, for our study, we have not established that, but then it is all, I mean, it's well known, like it's well reported that the specifically the tumor extracellular vesicles, they can cross the blood-brain barrier without even disrupting it by the mechanism called transcytosis. I mean, it's proven already. But then it seems like that was not true in our case, because in our case, blood-brain barrier was disrupted. And,
yeah, that's why I was curious. Yeah.
Yeah. That's a valid and good question to ask. And actually, we as of now we have not answered that question. But as of now, we have just established that blood-brain barrier was disrupted. It was not like the EVs were crossed. Was not just crossing because of transcytosis or some other mechanism, but the actual blood-brain barrier was disrupted. That's what, as of now, I can say.
And these were administered by tail vein or any other?
Yes.
Tail
Yeah,
vein.
they were
Sorry, I
they were
missed that.
injected. Yeah, they were injected. Probably I did not say that. So I was actually looking at the clock, so I should not pass 30 minutes. So yeah, so the EVs were injected tail vein, and it was a single injection here. And then after
wow.
24 hours, we did the analysis. I mean, we injected the sodium fluorescein dye, and then we did the analysis.
Wow, yeah, with a single injection, and this increase of the permeabilization is quite massive.
Yeah.
Wow.
Yeah, and even this one, the where we injected the C. elegans male transpected EVs, this was again a single injection.
Okay, thank you so much.
Yeah, sure. Thank you.
Thanks. We have another question in the chat box. Alex, if you want, you can unmute yourself and ask your question.
Okay. Hello, can you hear me?
Yeah, yes.
All right. So, very wonderful presentation. Thank you so much for sharing with us. But can you go back to the slide on your characterization of small EVs? I think I missed. Can you explain?
Characterization of small EVs. This one?
Yeah. This part. Yeah, this part. So, can you really explain this particular slide? I think I missed some points in this particular slide.
Oh, okay. Yeah, sure. So this is just these characterizations are like very well known. I mean, we did. Give me a second. I'm just unable to find my pointer. Yeah. So we did NTA analysis, which kind of shows you. It shows you the particle concentration as well, but we are more focused on seeing the size of these nanoparticles as like small, as I said, smaller extracellular vesicles, right? And are synonymously called as exosomes. So they are supposed to have size range, around 150 or below 150 nanometers. So we could see the, these are just the representative, graphs, and most of them were like the size was below 200 nanometer, or I would say like 150 to 100 to 150 nanometer, the average size. And then zeta potential, as these small extracellular vesicles, they are well reported to have zeta potential. I would say in the range of -10 to -30. I have seen. I may be wrong. There might be some other reports, but I have seen zeta potential of these small extracellular vesicles, not specifically prostate cancer secreted small extracellular vesicles. I'm just saying overall small extracellular, because of the surface charge, negative surface charge, they had a negative zeta potential. And then this is the tetraspanin analysis, which we did by flow cytometry, this CD9, CD60, and CD81, and it was found that 18.82% CD9 positive, 12.95% CD63 positive, and then CD81 was 62.66% positive. These small extracellular vesicles. So then we also did immunogold labeling by doing transmission electron microscopy. We found that CD9, CD63, and CD81 expression on these smaller extracellular vesicles. Yeah, I mean, we did some other characterizations as well, but these were related to the talk, so that's why I presented here these.
Oh, okay. Thank you. I think I understand it now. Thank you.
Sure. Yeah.
Thanks, Dr. Hilal, for a detailed presentation. I guess we have to move to the next section, so I will pass it to Shipra to start our career development time. Thanks again, Dr. Hilal.
Oh, thank you so much.